Here we have our green plant, but lately it's been struggling because its green leaves aren't attracting enough bugs to come pollinate it, and so its species could go extinct. So we want to genetically modify it so that it can start making red leaves, which will attract more insects. To do that, we will take a gene for red pigment from a strawberry. Here we have a red strawberry from which we want to extract the gene for red pigment. Here we have a cell from the strawberry, and we need a restriction enzyme to enter the nucleus of that cell to act on the DNA. Here it goes. Here within the nucleus, we see a section of DNA from the strawberry where the gene for red pigment is located. The restriction enzyme enters, cuts at both ends, and now we have isolated the gene that we desire. Here we have a plasmid from a bacterium. We need to insert the gene for red pigment into this plasmid. But first, let's magnify it to see it better. Now that we see it better, we realize that we need to remove the gene for green pigment and change it with that for red pigment, and we do that using the same restriction enzyme. Once again, the restriction enzyme enters and cuts at 2 points, and we have removed the gene for green pigment. This has left sticky ends at either end of the DNA in the plasmid. The sticky ends of the gene for red pigment are complementary to those found in the plasmid after the restriction enzyme has worked on it, so they bind together by the help of DNA ligase. So the result that we have is a plasmid with recombinant DNA, where we no longer have a gene for green pigment, but one for red pigment taken from the strawberry and put into a plasmid from a bacterium. However, we also need a marker gene. A marker gene is a gene we insert into the plasmid, in this case, to show us whether our desired gene has really been incorporated into the DNA of the organism we wish to modify. In this case, it is a gene for antibiotic resistance. Why? You'll find out very soon. So the same process is used as before, however with a different restriction enzyme, because it is a different gene. So the enzyme enters, cuts the DNA at 2 sites, and that gene is replaced with the gene for antibiotic resistance. Now that we have successfully modified the plasmid, we can insert it into a bacterium. Here it comes. Here we have a plant cell, and we can insert the bacterium which contains the modified plasmid into the nucleus of the cell. Now hopefully the DNA from the plasmid has been incorporated into the DNA of the plant cell. Now obviously a plant cell is not that large, so let's look at it in a more realistic size. To check whether the DNA from the plasmid has been incorporated into the DNA of the plant cell, we grow the plant cells on a nutrient medium with antibiotic present. The antibiotic, however, is toxic, and so only the plant cells that have successfully incorporated the DNA from the plasmid, including the marker gene and alongside it the gene for red pigment, only they will survive. The rest will not have gained antibiotic resistance and so will die. The plant cell forms clones by micropropagation, and these clones go on to form calyces, which are groups of cells, and eventually these will form plants. However, some of these plants will die or wither, as we said before, because the antibiotic is toxic to them, and they did not incorporate the gene that enables them to have resistance. The rest, however, grow to form fully functional plants. Here is our transformed organism, where it no longer only produces green leaves but also red leaves because of the red pigment gene that we incorporated into its DNA. And that is genetic engineering.